Tony Hunter, you are a professor at the Salk Institute. You have contributed a number of seminal discovery to science, and your work on protein phosphorylation is now classic. We were happy to welcome you for a special conference at Collège de France, and also a conference organized by students and supported by the Hugot Foundation. So you initially trained as a biochemist in Cambridge University, and then touched an incredible variety of biological topics. Is there some interconnection apart from the study of post-translational modification?
Well, right from the start, even when I was a graduate student, I was interested in many different areas. And then I worked on protein synthesis, which is important in many different systems. And that led me to work on more than one system, even as a graduate student. But it really was our interest in protein phosphorylation as a mechanism of viral transformation that led to, protein phosphorylation becoming a major theme in the lab, and because phosphorylation seems to regulate almost everything in the cell, that meant we could work on many different things. And I just like to work on more than one topic because it keeps me interested. If I kept doing the same thing day after day, I would lose interest, I think.
So when you have identified the tyrosine phosphorylation, did you realize the impact of this discovery?
I didn't really recognize it immediately. We first found it when studying a mouse virus that causes tumors. And quite by chance, I was also working on a second virus that causes tumors in chickens. And when that one turned out to use tyrosine kinases as well, then it suggested it could be important, widely important. But we really didn't know whether this was a virus speciality or whether it was important in the cell. And in fact, it was Renato Del Becco, who was at the Salk Institute, the Nobel Prize winner for his work on tumor viruses, who suggested to me this might be more important than I thought it was. So that was a nice touch. But it quickly became apparent that it would be widely important because not only were there other viruses that were shown to use this same system, but we learned that cells also have tyrosine kinases and that growth factors activate these kinases and is a mechanism for driving cells to grow. And so by that time, that was within about a year of the discovery, I think we realized it was going to be very important. And then finally, 2 or 3 years after that, the first human genes that were mutated in cancer turned out to be tyrosine kinases, and we thought, yes, this is going to be important. And it may even be possible to develop drugs that target these enzymes. And that didn't happen for a long time, but we wondered at the time whether that would be possible.
In fact, last year in the Collège de France, we watched an interview from François Jacob, and he said in this interview that when you discover something very amazing, he says first it's not true, second it's not new, and third it's not you that you discover this. And do you feel the same things or not?
Well, it turned out that other people had thought about tyrosine phosphorylation before I did. Fritz Lippmann had actually made the chemical back in the 1930s, so he'd thought about it then. And he even said it might be possible that tyrosine is phosphorylated. Then other groups had made it. There was a British biochemist who made and published about phosphotyrosine synthesis in the middle of World War II. I don't know what he was doing in the middle of World War II. And then someone else had reported that it's a storage compound in Drosophila eggs. They store tyrosine as phosphotyrosine because it's much more soluble. So in one sense, it wasn't new, but it was a surprise to the protein phosphorylation community because they thought that it could only be serine and threonine phosphorylation. And that's all in the 25 years that it had been worked on that had been found. And so when we stumbled on it, I called up as many experts as I could, and none of them said that they'd heard anything like this. So it was sort of new in that sense. It wasn't such a surprise. I mean, chemically, it was not an unreasonable reaction. And surprisingly, I mean, the community didn't, there was no objections to this. It was very quickly accepted by the community, unlike some radical ideas where there's a lot of opposition to it. In this case, it was very quickly adopted, and people realized how important it was.
So nice for you.
It was nice for me. I didn't have to fight to persuade people this was important, right?
So you made very strongly the point that on your recent studies on pol 3 driven transcription, that you can sometimes stumble across medically relevant issues when studying extremely basic questions. I think I would argue that you can also do the reverse. By studying very applied medical issues, you can also stumble across very basic mechanisms of regulation. So in this dialogue, in this cross-fertilization between the two fields, how do you think this can be promoted? And will we have to build research centers immediately adjacent to teaching hospitals, or should the connection be organized?
Yes, I mean, that's an interesting question, how one should do it. The Salk Institute is a standalone research institute. We have no clinical connections directly. And that has been a challenge for us, is to interact with clinicians who have medical problems where we might be able to collaborate and help them understand the disease. At the Salk, we've struggled with this. We've talked about recruiting clinician scientists. That hasn't really happened, and right now in the cancer area we're reaching out to the local UCSD Cancer Center, which is a comprehensive cancer center where they have obviously clinical research as well as basic research, and we're trying to get more closely integrated with them. In addition, we have just started at the Salk an effort to try and improve therapy for pancreatic cancer, which, as you know, is one of the most lethal cancers. And that came about through a national effort by Stand Up to Cancer, who wanted to fund so-called dream teams of scientists and clinicians located in different institutions in the country and bring them together for 2 meetings a year with the goal of doing some basic research and some clinical research to the hope was to be able to initiate new clinical trials within a 3-year period. And so that's been another mechanism. It's not local, it's dispersed, but it seems to have worked. I was surprised how well actually people integrated from different disciplines and from different institutions. And now that has been expanded to the United Kingdom. There's a team which has members both in Cambridge and London and in the United States. So that, I think, is another good mechanism. But I agree, having the institutions close by one another is a good start, but still somehow, I wouldn't say forcing, but persuading people they ought to interact is very important, and that's not always so easy to do.
Not at all. So you mentioned that hundreds of post-translational modifications will have been described, and how do you see the future of protein post-translational modification?
Well, I think people will continue to catalog the existence of these sites in different cells, under different conditions, in organisms, under different conditions. There was a very nice paper that just came out in Science where they used 18 fungal species to study evolution of phosphorylation. So I'm sure more of that sort of thing will happen as the technologies have become fairly routine now to, for instance, identify phosphorylation sites. I think there will be a lot of emphasis on, trying to understand how the process evolved, but obviously more importantly what it's used for and whether it's clinically relevant in terms of developing drugs, as has been the case in cancer so far. As I said, there are 36 protein kinase inhibitors currently in clinical use, most of those for cancer therapy. And as we heard later this afternoon, there are another several, probably 100 in clinical trials. So that's a nice intersection between basic research and clinical use.
With respect to the way you organize your research, I was very struck to see that you are using cutting-edge, up-to-date technologies, and at the same time extremely inspired research, really touching every bit in biology. How do you maintain this equilibrium and continue having fun in the lab, as you mentioned?
Right. Well, I run a lab. It's not a very typical lab. I recruit graduate students, but mostly postdoctoral fellows, and tell them they can do what they want. And so some of them come with interest they've already had in graduate school, or else they find something interesting. And then I expect them to develop their own projects. So that tends to take the lab in different directions. I mean, people obviously apply to my lab because they're interested in what we have done in the past. So that is phosphorylation and ubiquitination. So that's a sort of natural focus. But I had a postdoc who came and said she wanted to study the molecular basis of limb regeneration in axolotls. And she worked in the lab for 5 years, and we found some interesting things, and I found it fascinating, and so I encouraged her to do it. And even though I didn't have any grant funding, we found money somewhere. And
so freedom is the key.
Freedom is the key, and what's surprising is that often you start, or people start out on different projects, and then just sort of by chance they seem to intersect, and then people begin to collaborate. And so you never can predict the course of science, obviously. And so I've encouraged this because I think, it keeps me interested in different areas, and I keep learning. But also because that means that the postdocs can take their projects away with them to their new position. If they go to an academic position, then they can take that project with them, because we'll start something else in its place. So in that sense, it's not terribly focused, but I think it's been a successful way to run
extremely productive and,
yeah, right.
and
so
really mind opener.
We know that approximately 500 protein kinases are found in mammalian. Mm-hmm. And so do you know how many could become drug targets?
It's an interesting question. There were some studies that were published a couple of years ago about the human kinome, pointing out that 90% of papers published on protein kinases are on 10% of the kinases. So that means 90% of the kinases have not really been studied at all, maybe 1 or 2 papers. I think it's a case of people looking under the lamppost. They tend to work on things that other people have already shown to be important. And so there's a lot of room there for identifying protein kinases that could be important in disease. The fact that cancer genome sequencing methods have revealed so many mutations in kinases is a sort of an opening to begin to study some other kinases. The only issue there is that most of these are rather rare mutations. They're only found in a small percentage of 1 or 2 cancers. So they may not be a good drug target in that sense, but they're telling us something about the cancer almost certainly that might be targetable. And so I think, there's plenty of, as I said, plenty of room to begin to work on other kinases and hopefully find something important.
Why are you interested in simulation? I suppose it's not because it's popular or trendy.
No, it was one of those accidents. We were studying something else altogether in fission yeast, and my postdoc was trying to understand the Aurora kinase homologue and decided he wanted to do a 2 hybrid screen to see what proteins interacted with it and what proteins might regulate it. And he came across the one of the subunits of this RNF4 sumo-directed ubiquitin ligase, and on looking at the structure, it had a ring domain, and we'd already shown for other proteins that the ring domain was the key part of the ubiquitin ligase. We started working on Sybil as others were working on the ring protein in SCF. And so we thought it was probably a ubiquitin ligase, or could be. And then he noticed that at the N-terminus it had these short motifs, 4 short motifs that others had suggested might bind sumo, and so that's when we started working on sumo, and we guessed that the sumo binding motifs might be used to target the ligase activity to sumo-related proteins, and so we've sort of continued to work on it since then, but it was one of those accidents. We weren't deliberately thinking of working on sumo.
You showed very clearly in your Collège de France conference that the complexity of genes, maybe 20,000, is increased by alternative splicing, alternative initiation, and then that post-translational modifications raise the complexity to over 1 million. What do you think is the next step of complexity? Is it RNA, the RNA world?
Well, there's no question that the RNA world has greatly increased, you know, the possibilities of regulation in cells. I mean, microRNAs are obviously one very important area, and we know they regulate gene expression. But there are so many non-coding RNAs that have never been looked at that I think there's another whole world out there that remains to be discovered and is something for the next generation to work on. It turns out that the term RNA world was actually invented or coined by Leslie Orgel, who was a faculty member at the Salk. He was one of the founding faculty at the Salk Institute, and he was very interested in prebiotic synthesis, and he proposed that the first nucleic acid would be an RNA, not DNA, because it was chemically easier to make. And so he spent most of his career trying to develop methods under which RNA molecules could have been synthesized under prebiotic conditions. And so the Institute, even though it doesn't have an RNA biologist actually, since Leslie died, has been very interested in the role of RNA in evolution and cell function. So that's certainly going to be, I mean, lipids is another area that I think is a lot of complexity. How much is important we don't know, but now by mass spec you can see that every lipid like PIP2 has hundreds of possible variants. Now, do they do different things? Possibly they do. We just don't really know. And same with the glycome. You know, there are endless different combinations of sugars that are put onto proteins or just made as molecules. And now there are methods to characterize them chemically, and we're beginning to learn they probably do different things. So.
Thank you very much, Tony Hunter.
It's my pleasure to have
Thank you.
talked to you and also to have come to the meetings.
Thank you so much.
Yeah.